Assalamualaikum. Today, we want to present about plant transport for AGR499 crop science.
Hi, my name is Nurul Fatah Najwa binti Muhammad Haluzaki. My student ID 2024554479.
Hi, my name is Nurul Intan Naziha binti Ismail. My student number is 2024939151.
Hi, my name is Nurul Nazila binti Syarah Akbar. My matrix number is 2024745907.
Hello, my name is Nurul Quran binti Muhammad Khairi. My ID number 2024745473.
My name is Puteri Aisyah binti Adnan. My ID number is 2024746049. Transportation system in plant consists xylem and phloem transport. Water transport is the movement of water and minerals from the roots to the different part of the plants through xylem tissue. Sugar transport is the movement of the carbohydrate from leaf to the other tissue throughout the plant through phloem tissue.
For the pathway of the translocation, non-photosynthetic plant components also need carbohydrates, even though the plant produces them in their leaf through the photosynthesis process. Sugars move from the sources or area with an excess of carbohydrates to the sink, or we call it as the area where the carbohydrates are required. The movement of carbohydrate through the phloem from the sources to the sink, we call it as translocation. Main element of phloem are sieve tube component and companion cells. Sieve tube elements have no nucleus. Sieve tube element transport sugars and other organic compounds through the phloem of the plant. The phloem in the plants is a system that facilitates the efficient movement of nutrients through the special cell. We call it as sieve tube elements and companion cells. Sieve tube elements are elongate cells with large pores, allowing for the movement of sugars and other organic compounds. Companion cells, rich in organelles, support the metabolic functions of the tube elements and provide them with energy and enzyme. This system ensures that photosynthetic products are distributed through the plant for growth, storage, and other metabolic processes. Next is phloem transport. The tissue that transfer nutrients from the fully ground leaf to developing or storing organ and roots is called phloem. A highly specialized process for redistributing. The first one is photosynthesis product. Second, other organic compounds like metabolites and hormones. Third is some mineral nutrients. Redistributed from the first one is sources. Any exporting area that produce more photosynthesis than it's required, which means like the leaf. Second, sink is any organ that is not able to produce photosynthesis or that produce enough amount for photosynthesis to meet its own requirement, like flowers, fruits, and immature leaf. Next is about phloem sap. Sap in the nutrient and carbohydrate-rich fluids that pass through the phloem. Building blocks for the plant's actively growing section are provided by the translocation. Water, sucrose, other sugars, hormones, and acid amino are all present in the phloem sap. It moves from the site of production or storage of carbohydrates, which means photosynthesizing leaf are sugar sources, to the site of utilization like sugar sinks such as grains.
How are substances translocated in the phloem? Two steps in the translocation require energy, which is loading and unloading. Loading at source and transport of solutes from source into the sieve tube, while unloading at sink and the removal of solutes at sink.
The pressure flow model describes how sucrose concentration determines the direction of the flow. In this section, I will explain about the direction of the flow in the pressure flow models. Source cells produce sucrose by releasing gas into the phloem. The fluids begin to flow when the source cells pump sucrose into the phloem. When the concentration of the sucrose in the phloem increases, water will enter by osmosis. The turgor pressure within the sieve tubes increases when water flows into it. This pressure will force the sugar solution through the sieve tube to region of low pressure. The solution passes by sink cells and will take up sucrose as the sucrose concentration decreases. Water flows by osmosis out of the phloem to regions of high solute concentration. The water loss will decrease the pressure in the phloem and pull fluids from other higher pressure regions of the phloem.
Phloem loading refers to the first transport of glucose, amino acids, and other components from photosynthetic cells to conducting sieve tubes. Photosynthesis produces glyceraldehyde 3-phosphate, which is then transported from the chloroplast to the cytosol. At night, this molecule, together with glucose from stored starch, is transformed into sucrose. Both of these processes happen in mesophyll cell. Sucrose passes from the mesophyll cell to the sieve tubes to the sieve elements through the leaf tiniest veins. Sugars enter sieve elements and partner cells through a process known as sieve element loading. Sugars concentrate higher in sieve elements and companion cells than in mesophyll cells. Sugars are carried out from the source tissue through a sieve element and companion cell complex, which a process known as export. Long-distance transport refers to the movement of molecule from source to sink tissue.
How are substances translocated in the phloems? From photosynthetic mesophyll cells, sucrose follows a symplastic route into phloem parenchyma. Then the route may be apoplastic or symplastic to companion cells and sieve tube members. Then, solutes can move from mesophyll cells to the phloem by apoplastic or symplastic pathway. Sugars and other solutes produced in mesophylls can move to phloem by apoplastic pathway. Mechanism of subflow in plant vascular tissues. For driving force for bulk flow in xylem, transpiration from leaf, and for phloem, active transport of sucrose at source. For site of bulk flow in xylem, non-living vessel elements and tracheids, and for phloem, living sieve tube element. For pressure potential in sub for xylem, negative, and in xylem is positive.